Hi, my name is Paul Templier, and this is a presentation of our new article published at IEEE CEC 2024. This work is called Genetic Drift Regularization on Preventing Actor Injection from Breaking Evolution Strategies. It was a collaboration with Emmanuel Rachelson and Dennis Wilson from ISE Supero and Antoine Cully from Imperial College London. In this paper, we take the context of neural policy search, where policy is represented by an artificial neural network. This policy interacts with an environment in a sequential decision-making process. At every step, the policy sees the state of the environment. It chooses an action and gets a small reward. The sum of all rewards is used as the fitness score of the network in an evolutionary optimization loop, which will then generate a new, improved policy to be evaluated. More specifically, in this work, we're using evolution strategies, or ES, for the continuous optimization of the network parameters. The ES only uses the total score of each evaluation, but in the context of policy search, we have access to each sequence of a state, an action, and a reward. If we keep all of these transitions, we can then use deep reinforcement learning, or RL, to train a new policy called the actor policy. The genome of this actor is then traditionally injected into the population of the ES to inform the search. This is the process of actor injection. Actor injection is frequently used as part of a more complex evolutionary RL method, but we propose to study specifically here how it interacts with the distribution-based structure of evolution strategies. There are many complex variants of ES, such as CMA ES or CEM, but they all share the same core structure. They rely on a distribution in the search space, from which we will sample a population of solutions. These solutions are then evaluated and given a fitness score. This score is used to estimate the fitness gradient and update the distribution. From that distribution, a new population is sampled, and the loop repeats. If we focus on the update of the ES, this is the base mechanism. Once the population has been evaluated, we can compute the mutation vector between each solution pi I and the center of the distribution theta t. Each solution is given a weight based on its performance, which is used to scale the mutation vector. By averaging the weighted mutation vectors, we get the update vector for the ES center, and this gives us the new ES center theta t plus 1. Going back to actor injection, if we use trajectories to train an actor policy with RL, we have two solutions to keep track of in the search space, the ES center and the genome of the actor. If we measure the distance in the genome space between them, we can see that naturally they follow different paths, and they drift away from each other. This is a phenomenon we call genetic drift. The genome of the actor gets further and further away from the ES policy during the evolution. But what is the impact? If we take the ES update and add the genome of the actor far away from the ES, we can compute the mutation vector, which is large because of the distance. Even with a small weight, the mutation vector will be still large. The ES update vector will be impacted by the weighted sum, and the update direction will be biased by the actor, even if it's not performing well because of the genetic drift. We can project the trajectories into 2 dimensions for the ES in orange and the actor in gray. On the right is the fitness plot for both of them. They start from the same genome. After a few generations, they diverge and drift apart. The ES finds good policies while the actor struggles. After around 200 generations, the actor finally gets good and gets a larger weight in the ES update when it's injected. This has a big impact on the update, and we can see how the ES gets pulled towards the actor. Pulling the ES away from its path makes its fitness drop in orange. So the actor becomes the best solution in the update and keeps pulling the ES towards itself. Finally, the ES gets close to the actor and will just follow it to the end, never getting back to its original fitness. In this case, the injection of the actor broke the ES optimization instead of helping it. To fix this issue in the injection process, we propose a simple method called Genetic Drift Regularization, or GDR. When training the actor in TD3, we take the standard loss based on the evolution of the actor by the critic, and then we add a regularization term based on the genetic distance. In other words, we constrain the optimization to keep the actor genetically close to the ES. When looking at the genetic distance on the left between the actor and the ES, we can see the 2 variants of GDR in yellow and blue keep the distance low from the start, and there is no genetic drift anymore. On the right, their projected trajectories overlap, as they stay close to each other genetically during the whole evolution. By reducing the genetic drift, we reduce the negative impact of actor injection on the performance of the ES. When we look at the fitness of the ES on the left, the ES with GDR in blue and yellow can reach better scores than active injection in red, than other methods to fix injection in black. While we constrain the actor to say genetically close to the ES, symmetries and permutations in neural networks still allow it to find interesting policies, as we see with the fitness of the actor. Keeping the actor close to the ES can actually help it find better policies and learn, as seen on half theta in the bottom left figure. To conclude, in this paper we studied the actor injection mechanism in the context of evolution strategies. We highlighted the phenomenon of genetic drift, where the ES and the actor drift away from each other, and how it can negatively impact the ES. We introduced a simple fix with the genetic drift regularization method to preserve the performance of the ES against the potential negative effect of actor injection. As actor injection is often part of a more complex method combining evolution and reinforcement learning, we propose as future work to study how GDR interacts with such frameworks, such as CMASA. This was a summary of genetic drift regularization presented at IEEE CEC 2024. Please don't hesitate to check out the paper for more details or to contact us for any questions.